We're going to start these practice problems by looking at data from a test cross. This data shows us the frequency of recombination between pairs of alleles. We're going to try and determine the number of linkage groups, that is, how many of the genes are linked together and how many move independently of a larger group. Okay, so I'm going to draw down here 2 chromosomes. Well, we'll start with 1 chromosome. And if we start with this bottom cross, C and D experience recombination between the 2 genes 28% of the time. So let's draw them out on the chromosome. C and D are 28 centimorgans apart. B and D are 10 centimorgans apart. And then B and C are 20. Well, that makes me believe that rather than B being over here, B should in fact be between C and D. So we've got about 20 centimorgans here and about 10 centimorgans right here. Well, wait a minute. 20 and 10 aren't the same as 28. Well, first of all, it's pretty close, you guys. It's a pretty good estimation. Secondly, when we're looking over long distances, like 28 centimorgans, it's the longest distance we're carrying about here, it's easy to underestimate long distances. So that means these shorter distances, 10% recombination leading to 10 centimorgans distance, 20% recombination leading to 20 centimorgans of distance. These shorter distances are a better estimate of the actual distance between the genes, and then a longer distance between 2 genes which are further apart, it's easier to underestimate that difference. We're not going to talk about the logic behind that, but it's really because there can be double recombination events, but we won't worry about that in our 183 class at all. Okay, so we've got C, B, and D here on a chromosome, and then we've got A and D right here, experiencing recombination between them 50% of the time. Well, so should we put, A out here on the chromosome? I mean, we could, but as long as there's 50% recombination, all of these recombination frequencies mean that the genes are unlinked. They could be unlinked on the same chromosome. That means they're located far apart on the same chromosome, or they could be unlinked on 2 separate chromosomes. So we could draw A on part of this same chromosome. I'm going to erase it for now and draw it far away over here. So we could put A over here on the same chromosome and say that this is greater than or equal to 50 centimorgans. Or, given what we know here, it's reasonable to say that A is on a totally separate chromosome from C, B, and D. All right, let's work on this problem. We know that we have 2 genes, gene T and gene D, that affect the phenotype of cucumbers. T and D are located 16 centimorgans apart on the chromosome, so the recombination between them is 16%. Let's talk about the results of the cross which would occur between these 2 individuals. We have a warty dull fruit. Warty is coded for by a big T allele and dull by a big D allele. And then we have a smooth, glossy fruit. Smooth is coded for by 2 little t's and glossy by 2 little d's. Okay. We can see the chromosomes of these 2 cucumbers shown here. We have a dihybrid individual. big T, little t, big D, little d. And we have a homozygous recessive tester individual, little t, little t, little d, little d. We know that from this tester, there's only going to be one type of gamete made, little t, little d. But from the test cross individual, or rather the double heterozygous individual, we can have 4 different types of gametes made. But not all of those gametes will be formed in equal proportions. Take a moment and think about which of these 4 gametes are the recombinant gametes, okay? Which of these 4 here are recombinant gametes? Well, because we know already what the parental chromosomes look like, we can find those chromosomes down here in the gametes. This first one and the second one contain chromosomes that look just like the 2 parental chromosomes, whereas these right-hand gametes contain a different genotype or a different combination of alleles than what's seen in the parent, big T, little d, big D. So that means these right-hand gametes are the recombinant gametes, and the left-hand gametes are the non-recombinant gametes. Sometimes these are called the parental gametes because they look just like the chromosomes of the parent. Well, if we know which ones are the recombinant gametes, we should also be able to find the recombinant progeny. So let's look at the progeny on the right-hand side here. Which progeny contain this big T, little d, or little t, big D chromosome? Right here, we've got big T, little d, little t, big D. These must be the recombinant progeny. And let's double check the top progeny as well. It looks like we've got a big T, big D, little t, little d. Those chromosomes look just like the parental chromosomes, so they are non-recombinant gametes, or rather non-recombinant offspring. What about this second chromosome that each of the progeny has, little t, little d? Where did that come from? Well, it must have come from the other parent. So every offspring receives a tester chromosome, a little t, little d chromosome. I'll erase some of my scribbles now as we start to talk about the frequency of each of these different combinations of offspring and gametes. First of all, how frequent will we see these non-recombinant gametes versus the recombinant gametes? And then similarly, how frequent, how common will non-recombinant and recombinant progeny be? Well, we know something about the T and D genes already, and that is that they are 16 centimorgans apart on the chromosome, which means there's 16% recombination between them. And that tells us exactly how many of the gametes will be recombinant. 16% of the gametes will be recombinant, and 100 minus 16 will be non-recombinant. So that's 84% will be non-recombinant. That only tells us about the category. It doesn't tell us about each individual in that category. So if the category is composed of 2 possible gametes, the total of which occur at 16%, then each one of these gametes will occur at about 8%. And then for the non-recombinant gametes, it'll be 84 divided by 2. So 42% of the gametes will look like either one of these. We can apply those same numbers over here on our progeny. 42% of the progeny will be non-recombinant with this genotype, big T, little t, big D, little d. 42% of the progeny will be little t, little d, little t, little d. And then 8% of the progeny will be recombinant with this genotype, big T, little d, little d. And 8% will be little t, big D, little d. Let's try another problem thinking about recombination and predicting progeny and gametes. So we have a butterfly with a dihybrid genotype, and I'll redraw the chromosomes up here, big W, little t, and little w, big T. This butterfly is test crossed with little w, little t, little w, little t, a tester. In 65% of the gametes, there is no crossing over. In 35% of the gametes, there is crossing over. So before we go on to this bottom question, what does this central paragraph tell us about linkage of the genes? Take a moment, pause the video, and think about how far apart W and T are on the chromosome. Well, if we see recombination crossing over in 35% of the gametes, then W and T must be 35 centimorgans apart. Recombination occurs 35% of the time. Let's think about this bottom question. Examining 100 progeny, how many will have the genotype big W, big T, little w, little t? And let's draw out those chromosomes in another orientation. So we know that this bottom chromosome will come from the tester parent. What we really want to know is, does this big W, big T chromosome fall into the recombinant or non-recombinant category? We can figure that out by going up here to the parental genotype again. Do you see a big W, big T chromosome? I don't. I see a big W, little t, and little w, big T. That must mean we need recombination to occur in order to get big W and big T. The other recombinant gamete will be little w, little t. And our non-recombinant gametes will be big W, little t, and little w, big T. Now, the reason that I bothered to draw out all of those gametes is because we need to think about how frequently this gamete will occur. We know now that 65% of the offspring will not have crossing over, will be non-recombinant. 35% of the offspring will be recombinant. But that 35 represents the total of all recombinant offspring. Instead, we just want half of that to represent this big W, big T gamete. Half of 35 is 17.5%. If we return now to this bottom question, looking at 100 progeny, how many will have this big W, big T, little w, little t genotype? We can say 17.5%, or about 17 to 18 individuals from the 100 that we examined will have this recombinant genotype.